In this chapter 22, we're going to cover microbial diseases of the nervous system. First, a bit of basic anatomy of the nervous system. The brain and the spinal cord are grouped together in what we call the central nervous system, or CNS, and then notice that the nerves emanating out of the spinal cord together constitute what we refer to as the peripheral nervous system, or PNS. A key point right off the bat is that microbial infections of the central nervous system are not very common. They're relatively infrequent, but they often have serious consequences, and so we are going to talk about them in some detail. Now, delving a little bit further into the anatomy of the nervous system, there are two really key anatomical features that you need to understand. First of all, the meninges. That's meninges. Meninges are three layers that protect the brain and the spinal cord. They cover the brain and spinal cord. The outermost of those layers is called the dura layer. The next one in is the arachnoid layer. This includes the subarachnoid space, which contains cerebrospinal fluid, or CSF. And then the pia layer is the innermost layer. We're going to look at a diagram of the meninges and the cerebrospinal fluid in a moment and talk about their function in more detail. The second key anatomical feature that you need to understand is not the big bad breakfast, or BBB, but rather the blood-brain barrier, which is abbreviated BBB, blood-brain barrier. The blood-brain barrier is a set of specialized capillaries in the brain. Now, capillaries, remember, are the smallest blood vessels that deliver oxygen-rich blood to a particular tissue in the body, but also the smallest blood vessels that harvest oxygen-depleted blood and return that blood into circulation. You have capillaries throughout the tissues of your body, but in the brain, the capillaries are specialized to prevent most large molecules from moving through them into the brain. For example, bacteria, red blood cells, and water-soluble molecules cannot move through, most of them cannot move through the blood-brain barrier, these specialized capillaries, into the brain. However, some entities can pass through the blood-brain barrier. Critically, of course, is oxygen. Your brain needs oxygen, and that oxygen can move from red blood cells through the blood-brain barrier into the tissues of your brain. Glucose, which also is key for feeding the cells of your brain, amino acids, white blood cells, which is key for your immune system to be able to protect the tissues of your brain, anesthetics, and does anyone know that last one? Alcohol, ALCOHOL. Alcohol can move into the brain. Maybe you're aware of this. The meninges and the cerebrospinal fluid are depicted here in this figure from your textbook, figure 22.2. So notice, first of all, on the top right there, this inset pattern or diagram shows the skull bone on the outside, and underneath is the three layers of the meninges: the dura layer, or outer layer, the arachnoid layer, under which is the subarachnoid space and the cerebrospinal fluid, and then the pia layer, which is the inner layer. Now, interwoven there are blood vessels that are part of the blood-brain barrier. That blood vessel pictured there is a capillary, but it's not any old capillary. It's a capillary that has modified periphery to prevent certain molecules from being able to move out of your blood into the brain tissue, and to allow other materials to do so. Now, if you look at the inset on the bottom right, you can see another illustration of the meninges. Here, the layers are the same, but their thicknesses are different than they are around the brain. So around the brain, the layers of the meninges are thicker for protection and cushion of the brain. Around the spinal cord, the layers are the same, but they're mostly a bit thinner. The dura, the arachnoid, the cerebral spinal fluid space, the subarachnoid space, and then the pia layer on the inside. So where is the blood-brain barrier again here? Well, it is in the capillaries that are in contact with the brain. Those are modified capillaries, and the cell walls and periphery of those capillaries is what prevents certain molecules from moving out of your blood into the brain. Okay, now a couple of notes on some of the diseases of the central nervous system. Several diseases that I want you to understand are inflammation diseases. Inflammation is something we talked about briefly previously, but to remind you, it's a natural response by the body involving increased movement of plasma, which is the liquid part of blood, and white blood cells to damaged or infected tissue. Now, inflammation is sometimes helpful in fighting infections, but when it happens in the brain or the meninges, it can be dangerous. Meningitis is inflammation of the meninges due to an infection, and there are different versions of meningitis. Bacterial meningitis is caused by bacteria. Viral meningitis is caused by viral. Bacterial meningitis is not as common as viral meningitis, but the consequences can be more severe. Encephalitis is inflammation of the brain. The CEPH part of that word refers to the head or brain, and inflammation of that region is encephalitis. In general, itis, I-T-I-S, refers to inflammation. Thus, meningitis is inflammation of the meninges, encephalitis is inflammation of the brain. Meningoencephalitis is inflammation of both. Okay, now a few specific notes on bacterial meningitis, which I mentioned a minute ago is not as common as viral meningitis, but can have really severe consequences. This disease is characterized by initial symptoms of fever, headache, and stiff neck, followed by nausea and vomiting. In more severe cases, if not treated right away or treated unsuccessfully, this disease can progress rapidly to convulsions, coma, and even death. Symptoms of this disease are caused by endotoxins or cell wall fragments of bacteria in the cerebrospinal fluid. And these toxins are what cause inflammation of the meninges. Treatment of this disease is administering antibiotics. This is a dangerous disease, and the mortality rate varies among different age groups and among different kinds of bacteria, but it can be up to 80% in the most severe cases. Diagnosis of this disease involves analysis of bacteria to find out what kind of bacteria are involved, if any, by examining the cerebrospinal fluid. And for this, you need to use a technique called a spinal tap. And I'll show you a diagram of that in a moment. Spinal tap is also referred to as a lumbar puncture. And in this procedure, a needle is inserted usually between the third and fourth lumbar vertebrae, as shown in the inset diagram on the bottom. Now, this is obviously a sensitive procedure where the doctor needs to make sure not to penetrate with the needle too far, because it could result in damage of the spinal cord. But assuming that the needle is penetrating just into that subarachnoid space where the cerebrospinal fluid is, then the sample of the cerebrospinal fluid can be extracted and examined for populations of bacteria. In a healthy person, you should not have bacteria in your cerebrospinal fluid. If there's bacteria in there, then they are the likely source of this disease. Now, one version of a meningitis, bacterial meningitis, is referred to as pneumococcal meningitis. Pneumococcal meningitis is caused by a specific bacterium, Streptococcus pneumoniae. Streptococcus pneumoniae is going to come up in multiple lectures now before the end of the semester, and when it's involved, we're going to sometimes refer to that disease as pneumococcal. The pneumo part comes from the species name Pneumoniae, and the coccal part comes from the Streptococcus genus name. So pneumococcal refers to Streptococcus pneumoniae. So pneumococcal meningitis is caused by that bacterium. This is the most deadly of the bacterial meningitis diseases. It can have an estimated 30% mortality rate in children, 80% in elderly people. An interesting fact, though, is that up to 70% of humans are healthy nasopharyngeal carriers of this bacterium. This means that you don't have meningitis, but you do have this bacterium in your nasal and pharynx passages. The problem comes when this bacterium makes its way somehow into the cerebrospinal fluid, which under normal circumstances does not happen. But when it does, it can cause a serious disease. This disease is most common in children from about 1 month to 4 years of age, and it can be prevented by vaccination. Now, I want to mention a couple of other key bacterial diseases of the nervous system over the next few slides: tetanus, botulism, and leprosy. Tetanus is a disease caused by a Clostridium species. This is a bacterium, specifically Clostridium tetani. We've encountered Clostridium before. This is one of the genus names that I wanted you to know for the last exam. This is a gram-positive bacterium. It forms endospores, and it's an obligate anaerobe, so it cannot live in the presence of oxygen. This bacterium is especially common in soils that have fecal animal wastes in them, and where this disease happens is in deep puncture wounds, usually when soil has been driven in. So if you step on a dirty nail or run into a rusty piece of barbed wire that has some soil on it, you may get this bacterium punctured deep into your skin where there's a lack of oxygen, and thus that bacterium may thrive in there. What happens is that when the bacterial cells are killed by your immune system, they release a toxin called tetanospasmin. And this toxin is a neurotoxin. It interacts with your nerves, and specifically it blocks relaxation pathways in your muscles. What this means is that your muscles contract and they cannot relax again. This disease can be prevented by vaccination with something called the tetanus toxoid. Tetanus toxoid is an inactivated tetanospasmin toxin. Tetanospasmin toxin is the toxin from the bacterium, but if we take a version of that and inactivate it and then inject it into a child or a young person, then that toxin will be, in the future, will be recognized by your immune system. You will have antibodies for that toxin, and it will be destroyed as soon as it's detected by your immune system in the future, and you won't suffer the consequences of that toxin. Treatment of the disease, if the disease sets in, is with antitoxin. Antitoxin, now you need to make sure to not get this mixed up with tetanus toxoid. Antitoxin is a blood serum that has antibodies in it. So the tetanus toxoid can be used as a vaccination as a preventative measure. That's in the previous bullet point. And that last bullet point here, I'm talking about if you already have the disease, how do we deal with it? Well, if you have the disease, we can help by injecting antibodies, and those antibodies help your immune system to target the toxin and destroy it. Now, think a little bit more for a moment about the potential consequences of this disease. If your muscle pathways, if the relaxation pathways in your muscles are blocked and your muscles cannot relax, what's going to happen to your body over time? It's like an increasingly progressively worse cramp throughout your body. And this is not only muscles like your biceps and your abs and your quads, but think about all the muscles that control the organs in your body. If those cannot function properly, you're going to start suffering organ failure and die. And that's eventually what happens if this disease is not treated promptly and properly. This is an old drawing of an advanced case of tetanus. You can see this person's muscles, almost every part of their body, you can see their muscles are contracted. Their fingers are curled up, their toes are curled, even their lower back muscles are tightened, causing them to arch. But even worse, their internal organs, the muscles controlling them, are starting to cramp as well, which is going to ultimately lead to the death of this person. The second non-meningitis disease that we're going to talk about is botulism. Botulism is also caused by a Clostridium bacterium. You should see a theme here. Clostridium bacteria, in addition to what you knew about them before, they produce neurotoxins that interact with your nervous system and cause diseases. So botulism is a disease that is a type of food poisoning. This is caused by a bacterium species in the genus Clostridium, but not tetani. This is Clostridium botulinum. B-O-T-U-L-I-N-U-M. This is also a gram-positive endospore-forming obligate anaerobe, just like Clostridium tetani. But it has different habits. This bacterium likes to live in food, and especially in food where there's no oxygen. So canned food, for example, if you don't sufficiently sterilize canned food or create conditions that prevent the growth of this bacterium, it will thrive in your canned food. And in your food, it will release a toxin into the food called botulinal toxin. And this toxin is also a neurotoxin, but this has the opposite effect of the tetanus toxin. This toxin blocks the release of neurotransmitter, causing flaccid paralysis. This is the opposite of what happens with tetanus. In this case, rather than causing your muscles to be permanently cramped, it causes your muscles to be permanently relaxed. And for the function of internal organs, this is equally devastating. Your organs need muscles to both contract and relax. Picture, for example, the muscles that control your lungs. You need them to contract and relax to breathe in and breathe out. If those muscles cannot contract or cannot relax, you're in trouble. Prevention of this disease involves, first of all, proper canning methods, sterilization, and acidity to prevent the germination of Clostridium endospores. Nitrites are a particular class of chemicals that can be added that prevent bacterial growth, and these are especially commonly used in sausage. S-A-U-S-A-G-E, that's a food that used to have big problems with botulism. In fact, botulus, in Latin, means sausage because this disease was named after sausages. Botulus is the Latin word for sausage. Now, I've said that it's a kind of food poisoning where what happens is that you ingest the toxin from the food. In fact, in regular botulism on the previous slide, it doesn't matter if you eat the bacteria. What the problem is eating the toxin that's in the food. However, there is a form of botulism that happens in the body, and this is infant botulism. This results from Clostridium botulinum growing in the intestines of an infant. Now, think back to the discussion we had previously in our last lecture. Why do babies have problems with certain diseases that adults don't have problems with? Well, in a lot of cases, it's because those babies lack the full complement of normal microbiota. Remember that in the gut, your normal microbiota help to fight off infections. Well, this is one example. If babies have their normal full complement of normal microbiota, including E. coli and many others, those bacteria dominate and they prevent infections by things like Clostridium. But a baby that does not have a fully complete normal microbiota may be susceptible to Clostridium botulinum actually growing in their gut and then producing the botulism toxin inside their body. And this has the same consequences for this infant as it would if you were ingesting the toxin. So this helps us answer the question of why not give honey to babies? Well, babies less than a year old generally don't have a full complement of their normal microbiota, even if they've come through a standard birth. And honey has Clostridium endospores in it. Most of the time that's not a problem because our immune system and our gut biota and the acid in our stomach all help us avoid any kind of infections from those endospores. But babies are more vulnerable. And why are infants more susceptible than adults? Well, we've already talked about that. It's because most infants lack the full normal microbiota of the gut. Another kind of botulism that's a little different, that happens in the body rather than in your food, is called wound botulism, and this results from the growth of Clostridium botulinum in wounds. This is more similar to tetanus in how it happens. In tetanus and in wound botulism, this is the growth of a Clostridium bacterium deep in a wound, and the reason it's in a wound, able to grow in the wound, is because of a lack of oxygen inside that tissue. The treatment for any kind of botulism is antitoxin, which we talked about with tetanus as well, and then supportive care to allow your nerve endings to regenerate. So review what an antitoxin is by going back to the tetanus slides when you're studying. Botulism can be diagnosed by toxin ID, so you can extract a blood sample and use chemical analysis to determine the kind of toxin that's in the blood. Okay, a third disease of the nervous system besides the meningitis diseases, the inflammation diseases, is leprosy. Leprosy is also called Hansen's disease. This is caused by a bacterium in the genus Mycobacterium. This is Mycobacterium leprae. This is a bacterium that grows optimally at 30 degrees Celsius. And now, what is your core body temperature in Celsius? Think about that for a minute. That's 37 degrees Celsius. So this is cooler than your core body temperature. This is not a bacterium that grows well in your gut. It grows well in the periphery of your body where your tissue is a little bit cooler, about 30 degrees Celsius. Peripheral nerves and skin cells. That's where it likes to grow. Transmission of this disease among people requires prolonged contact with an infected person. It is not a very contagious disease. You can't catch it by them coughing into the air and then breathing it in or anything like that. Even a brief touching, you probably won't catch it. It requires prolonged contact. Interesting minor note here, there is another way that you can get this disease. The human version of leprosy can be transmitted by armadillos. So if you're in the habit of catching and chopping up armadillos and eating them or stewing them up for an interesting weekend meal, this is probably not a good idea because you could get leprosy from them. Tell your friends. A couple more notes on leprosy in terms of the manifestations of the disease. Well, one form of the disease is called tuberculoid or neural leprosy, and this involves a loss of sensation or numbness in skin areas where the infection is happening. This occurs in people with healthy, normal immune systems, and sometimes it goes away spontaneously without any treatment. It can definitely be treated with antibiotics. Lepromatous or progressive leprosy is worse. This results in disfiguring nodules over the body. This usually happens in less healthy people who have weakened immune systems. Here's an example of a leprosy lesion from the less severe kind of leprosy, the tuberculoid or neural leprosy. Just a patch of numb skin, and this might clear up on its own, definitely clear up with the right antibiotics. Here's an example of lepromatous or progressive leprosy, which can result in nasty lesions, growths, and also ultimately loss of tissue from the body. Okay, now a couple of notes on viral diseases of the nervous system. I'm going to mention briefly two examples, polio and rabies. Polio is short for poliomyelitis. This is caused by the polio virus, which is transmitted in the fecal-oral cycle. That's a bit of terminology I want you to become familiar with. We've talked about that a little bit already. The fecal-oral cycle means that the intake of the disease is through the mouth into the gut. That's how you catch it. And how you pass it on is through your fecal matter into the waste stream. And generally, these kinds of diseases are problems where when sanitation is not so good. Most cases of polio are asymptomatic or have limited symptoms. So you, in fact, may have had polio and not known it. Maybe you had a headache, fever, sore throat, and nausea, thought you had the flu. Maybe you had polio. However, in a small percentage of cases, less than 1%, the virus leaves the lymph nodes and enters the central nervous system, where it damages the upper spinal cord, resulting in some amount of paralysis. This disease appeared suddenly in the US in 1894. Now, why is that? Well, before then, infants were usually exposed while they still had protection from maternal antibodies. Maternal antibodies would be antibodies that are passed from the mother to the baby while they're still in the womb. And those antibodies can provide protection to babies. However, improved sanitation actually led to delayed infection until children were more susceptible. So this is kind of counterintuitive, but when sanitation improved, there was less common polio infection, and so infants often were not exposed to the virus. Kids were exposed later when they no longer had protection from their mothers, and so then from their mother's antibodies that resided in their body for a little while, and thus they were more susceptible to the disease. Eventually a vaccine was developed which largely eradicated this disease from the US population. And that is how we're trying to eradicate this disease worldwide with vaccination. This is an example of how well the vaccination has been working. This is a graph showing worldwide annual incidence of poliomyelitis since the early 1980s. And you can see it's been a steady decline over the course of the last 30 plus years, to a point where there are very few cases worldwide. In 2007, there were less than 2,000 cases. All right, now the second viral disease I want to mention is rabies. Rabies is caused by the rabies virus. It's transmitted by animal bites. That's bites by non-human animals to humans. Thus, this disease is an example of a zoonosis carried by a non-human animal. One form of rabies is called furious rabies. These animals exhibit symptoms of restlessness and then are highly excitable. This is probably the more sort of famous or stereotypical version of rabies that you might think of. However, perhaps a more common form of rabies is paralytic rabies. In this disease, animals actually seem kind of woozy, unaware of their surroundings. As a result, these animals may seem tame, even approachable more than normal. You might find a raccoon on your front porch that seems like it's acting friendly. This would be a dangerous situation because you might be tempted to approach it and see if it's okay, and then it might bite you and give you rabies. Rabies is actually quite uncommon in humans. Occasionally it's transmitted to humans from animals, but it's more common in non-human animals. The way this infection works is, go to step 1 at the bottom of this diagram. The virus enters your tissue from the saliva of a biting animal. It's carried in the saliva of an infected animal. The virus then replicates in your muscle near the bite. In step 3, the virus then moves up your peripheral nervous system to your central nervous system, the spinal cord and brain, ascends the spinal cord, and reaches the brain ultimately, where it causes eventually fatal encephalitis if the disease progresses this far. Now, in the meantime, the virus is also entering the salivary glands and other organs. So 5 and 6 might be a little bit out of order here, because if this disease is to be transmitted, it needs to enter the salivary glands before the victim dies, so that the victim has a chance to bite and transmit the virus. Now, rabies is really a serious problem for humans if you let it get this far to where it gets to the brain. Rabies can be treated effectively if it's caught early. This graph shows the reported cases of rabies in animals throughout the USA. Note, rabid bats are found in almost all states, so they're not color-coded here. Rabid bats are fairly common throughout the United States. And then other animals are shown here. In the far east of the U.S., raccoons are more commonly reported carrying rabies. In the central U.S., it's more commonly skunks. And then there are a few areas where foxes are also important carriers. Now, the fact that Mississippi is in white here suggests that raccoons and skunks are not very common carriers, probably just bats. You can see here, throughout the U.S., raccoons are overall the most common carriers, followed by bats and skunks, and then other animals. Prevention of rabies before exposure can be accomplished with the injection of a vaccine. This is only really used with high-risk individuals like veterinarians who might have to interact with rabid animals, or wildlife control employees. And then post-exposure, vaccine plus rabies immune globulin are used in concert to try to prevent progression of the disease. The vaccine works how a normal vaccine does, which is a deadened version of the toxin that helps to prime your immune system to recognize that toxin and attack it. But then the rabies immune globulin is blood serum with rabies antibodies, so that the antibodies can tag the toxin for destruction by your immune system without your immune system having to create these antibodies and go about doing the tagging. The antibodies are injected into your body and gives your immune system a leg up.